have also been working on some additional solutions in the project, and I will talk about the artificial biofilms that we constructed ourselves. And also Alejandro will present in the next talk some other solutions. So we started from our experience from other environments where we worked in the past, and to our knowledge, basically any surface, any material that you will put in the environment will get populated one way or the other, and it's really hard to stop this process. If there is if you put the metal into the environment, there is also some other properties of the material that affect how this population process will develop. If I just show you a few examples, I know that all of you, or some of you, have a lot of knowledge about the metals, maybe also the concrete. These two examples always develop biofilms, natural biofilms in the environment, but also other materials are more or less the same. If you go to your home, you know how difficult it is to remove these natural structures, let's say, once they have developed. Also in your body, the same thing, the skin, the tooth, the gut. Also, if you have an infection, it can develop in the heart, and so on. We call this, we think it's a very well programmed process, and if we focus on the metal, it's very important how these steps progress, how the succession of this natural structure happens, and it's very important maybe the yellow cells that stick to the surface. Which are those, and what are the ones that come next, and then the third ones that attach, and so on. A lot of literature is already available about this, but the most important thing is, can we mimic this? Can we do something similar? So can we build a structure, maybe in the end, this one here, not so complex, of course, because in the natural environment it's really diverse and also very stable, but can we build something similar? So we say there is an eco-evo approach, which is the ecological evolutionary based approach, where you can use insights from the ecology and evolution to build or develop some tools with which you can build a structure that will enable a coexistence of different species, and they will stay in mutual relation, be very stable, and try to stop other unwanted organisms from entering this structure. It's maybe something like the river. It's an analogy. The river will never stop flowing. Also, the biofilms, the natural biofilms, will never. You cannot really stop them from forming, but you can say which way the river will flow. You can also steer the communities that form on the surface one way or the other by what you put on the surface. Two models maybe are important here, and in both cases the idea is you have to focus on what sticks where and what comes first. From current knowledge, I think you can say that certain species are really important to come first, and then the next species that follow, basically the first comers will affect the ones that are coming next and how the structure will form. And if we know this, can we put something on the surface that will prevent the next coming species that will come? What we're faced with is an enormous diversity. This is what we also saw in this project. If you do not grow, if you don't cultivate the bacteria, the diversity is huge, especially in the seawater, especially in March, as you also pointed out. But what we will put on the surface will have to fight against all those microorganisms and select them maybe the way we want. So A and B candidates or others, of course, will interact with all the microorganisms coming from the environment. So we should help them out a little bit. Maybe by protecting them. We have built a capsule. It can be a microcapsule or a nano-sized capsule to protect the surface of the bacteria against grazing and against competition. And also on the other side, use the organisms which will increase their competitive advantage in the structure that we are building. So to use something that produces antimicrobial compounds that will stop the growth of unwanted ones. Finally, the solution was a multi-layered structure, which you can put on basically any coating or raw material. There is an alternation of, let's say, polymers and cells which are encapsulated, which are protected, which produce antimicrobial compounds. And you can build this structure layer by layer, the way a capsule is built around a cell layer by layer. So the same approach. This is more or less the idea, and just some primary results, but also our colleagues will show more results on the testing that was done in the ecological part, ecotoxicological examinations. So the idea here was to expose this structure, expose this biofilm, which is on top of the closing coating on the material, on the metal, expose it in the environment and try to see if there will be any shift in the community which will develop on the surface. So some species will attach, some will not. And there is just a simple DGG analysis of the community on the surface. If there is no biofilm on the right, number 2, and if there is an artificial structure on the surface, you will see a shift of this community that has attached. So these are just the first steps of the species, species that are coming to the surface, not the macro organisms that will stick later on. So this shift we have seen, and also we're waiting for the results from the sequencing center to try to identify the species that are involved in this case or the other. Also, some ecotoxicological tests were done. Gabriele will show the results. It is important if the structure is stable, if there is any release of the components, if they are toxic, and so on. And with this, I can conclude and maybe answer a few questions.